All right. We are starting
Okay.
the next 1 minute.
Okay, sir.
So at 10 we'll begin.
Yeah, why will I? What happened? So many things happen. Yeah.
Okay, Matthew.
Yes, sir.
The lecture is going to begin now. So you
Okay,
can, if there is any noise from your background, you can mute your microphone. So when you have a question or something like that, you can put on your microphone and then we'll discuss.
Okay.
So good morning, class. My name is Dr. Nafiu Lawal, and I'm a registered veterinary surgeon. I have a master's in microbiology and a PhD. Today I'm going to be your facilitator for the first NSC211 microbiology and parasitology. This course, Matthew, can you put off your microphone? Can you mute yourself?
Okay, sir.
Thank you. So we are going to have our discussion, our first class, NSC211, medical microbiology and parasitology. And the whole idea about this course is to expose you as nursing students on the basic concepts that you are supposed to know in terms of microbiology and parasitology as you care for your patients. As you all know, microbiology is the study of microorganisms, meaning those organisms that you cannot see without the aid of a microscope. So we are going to go on a journey where we will see that microbial wall and see so these are the learning objectives that we want you to know. We want you to be able to define the term microbiology. Matthew, is it from your end? Matthew, is it from your end? I said you should mute your microphone. You should mute your microphone. Can you see the icon for your microphone?
I'm not seeing, sir.
Just place a cursor on the WhatsApp this thing. You can see an icon there that shows that it is your microphone. Can you see my screen?
Yes, I'm seeing, sir.
So did you see where I just placed my cursor where it says mute? It gives a picture of a microphone and it says mute. So if I want to mute myself now, I will just press on it and I will become mute. Okay. So the essence is when you do that to your own hand, so you can see now you are muted. Okay, thank you. Now, like I said, the learning objectives that we want to achieve by having this course is we want you to be able to define the term microbiology at the end of this course, and you should also be able to group organisms and know their classification. You should also be able to distinguish between what are prokaryotes and what are eukaryotes, and you should also be able to explain the distribution of these microorganisms into the basic various domains that they are distributed into. And we also want you to be able to state the characteristics of these microorganisms that are there in each domain, and also be able to state the characteristics of viruses. Okay, so these are the 6 intended learning objectives that we want you to be able to achieve. So the first unit, if you have if you have downloaded the courseware, the course material from the noun e-courseware, you'll be able to follow me. I already gave you from the timetable and lesson plan, you should be able to know the various units that we are going to discuss, and I am expecting that you have already started reading the material, so you will be able to understand what I'm trying to say. Okay. Now, the first unit is this. It is all about what are microorganisms. Uh, like I said, microorganism is the microbiology is study of microorganisms, which are those organisms that you cannot be able to see without the aid of the microscope. So because it is because that deals with these organisms. These organisms comprise of bacteria, fungi, viruses, and protozoans. Even though in protozoans, some of the organisms in protozoans sometimes some of them can be seen with naked eye. But when you talk about bacteria, we talk about fungi, you talk about viruses. For viruses, you cannot even see them with the aid of a compound microscope that we are familiar with in most of the laboratories that we operate in hospitals. You need sophisticated microscopes such as electron microscopes, the various types. We have the transmission electron microscope, we have the scanner electron microscope. These are the type of microscope that you'll be able to see viruses with. But in the case of fungi, some of the fungal organisms can exist in two forms. There are those that are existing in the form of hyphae, the mold that we are familiar with. That one we can see with our own naked eyes. But for fungal organisms that exist in two forms, the yeast type you cannot see without the aid of a microscope, because it is assuming that shape and size similar to bacterial organisms. So for the bacterial organisms, the largest bacterial organism is about 1 micrometer, and that is beyond the same capacity of the human eye. Okay, so uh, for, for bacteria, the yeast type of fungi and viruses, you need the aid of microscope for you to be able to see them. While for protozoans, some of them you can see with the use of naked eye. Like for example, amongst them, among the protozoans, we have those that are actually parasites. Like for example, liver fluke, tapeworm, and beefworm, the nematode organisms. All of them, they are organisms that are classified under protozoans. They are actually there is a branch of plastology called helminthology, where these organisms, which are multicellular in nature, are normally discussed, and we'll see them later in the course as we progress. So most of these bacterial organisms and the fungal organisms, they are made up of cells. Like we know, in our basic biology, we were informed that the fundamental unit of life is called a cell. So for these organisms to exist, there are those that exist as single cells, and there are those that exist as multicellular organisms. So for bacterial organisms, all of them are unicellular in nature. It means that it is a single cell which has the capability, which has the capability to exist as an independent organism. A single bacterial cell can exist independently. It will undergo its own metabolic processes. It can do all those Mr. Niger plus D that we know, meaning that it can grow, it can move. You know, there are some bacterial organisms that have structures that will aid them to move, and it can also respond to stimuli when there is an environmental condition that is favorable to it. It will move toward that particular environment, and when the condition is favorable, it will move away and stops like that. Okay, so that is for bacterial organisms. But for fungal organisms, the it is only the yeast form that exists as a single cell. Okay, it exists as a single cell. But others, they exist as multicellular organisms. Okay, they exist as multicellular organisms. Now, Since we said that these microorganisms are fundamentally made up of cells, either in singular form or as multicellular, now what are those cells? In basic biology, we know that we have two types of cells. There are cells that are called prokaryotic cells, and there are cells that are called eukaryotic cells. Now, the major difference between them is based on their own structural complexity. For example, for prokaryotic cells, there are certain fundamental so can you hear me now? You can all hear me, right? So like I said, we have two types of cells, the prokaryotic cells and the eukaryotic cells. And the differences in them is based on their own structural complexity. The major difference between them is that in prokaryotic cells, they do not have a membrane-bound nucleus, and they also don't have membrane-bound organelles. But in the case of eukaryotic cells, they have a true nucleus, which is bounded by a membrane, and there are also organelles that are present for you to see. So that's the major difference between prokaryotic cells and eukaryotic cells. Okay, so fundamentally, bacteria is an example of a prokaryotic cell, when while the fungi usually and plants, they are examples of eukaryotic cells. So this is the example that we are talking about. On my left here, on the left of the screen, you can see a typical bacterial cell. If you look at that part of the bacteria that is labeled as nucleoid, that nucleoid is the genetic material of the bacterial organism. It is supposed to be what the bacteria possess as the whole genetic makeup of that bacterial organism, and it is what the offspring of that bacterial organism will inherit from it. So normally, from our basic biology, we know that the genetic material is inside nucleus. That is in eukaryotic cells. But in the case of bacterial organisms, they have their genetic material just inside the cytoplasm, without bounded by any membrane. Okay, so that's why you see it as a coily structure like this, that are spreaded, or that are occupying a very huge area of the cytoplasm. Okay, and they also possess a capsule, which we don't. In the case of eukaryotic organisms, in the case of eukaryotic cells, there is no any capsule that is there, and they also possess cell wall. In the case of eukaryotic organisms, it is only plant cells that possess cell wall. In the case of animal cell, we don't have any cell wall. But in the case of bacteria, apart from the cytoplasmic membrane that we all possess, they also possess cell wall. Even though the composition of the cell wall differs the bacterial organism into gram-positive bacteria and gram-negative bacteria based on the amount of peptidoglycan that they possess. Okay, like I told you that some bacterial organisms can move. They are motile. They use what is called flagella. as a single cell, it can still respond to stimuli using the flagella. So they use flagella, to move away. There are different types of flagella. When you have a single flagella like this, protruding from one angle, there is a name that you can call that type of flagella. Call it monotrichous flagella. In some cases, just like you see the pili, this pili the bacteria is using it for it is using it for attachment to surfaces. So because of that, hello Helga, you are messing up with our screen. You're writing something, right? Hello, is it Helga? Somebody just wrote on my screen. Okay, so can you please all mute yourselves? Thank you. Now, so that's the difference between them. In the case of eukaryotic cells, you'll find that they do not have any flagella or pili. Eukaryotic organisms like who is Hilda, please? You're writing on my screen. Okay. So in the case of this bacterial organism, they can use pili. There are certain functions that they can use the pili to do. Like I said, they can use it for attaching to surfaces so that they become firmly attached to surfaces, and that way they will not be easily dislodged from wherever they are attaching. And they also use it for sex, meaning that they call it sex pilus. It's not sex in the actual sense, but rather they use it for genetic transfer of materials between gram-negative organisms. So when a particular gram-negative organism possesses a kind of resistant trait against one antimicrobial substance, it can use the sex pilus to transfer that resistant trait to other gram-negative organisms so that it will help them also to become resistant to that particular organism or to that particular antimicrobial substance. Now, if you look at it now, in the case of eukaryotic organisms, or in case of eukaryotic cells, they have a predefined nucleus where the genetic material is inside the nucleus. And the genetic material there is the chromosome. And you can see it here having that X shape, that we are all familiar with. Okay, and as you can see, there is a definite nuclear membrane that the nucleus is being surrounded with, and you can see various membrane-bound organelles such as lysosome. You can see ribosomes. You can see Golgi body. You can see peroxisomes. You can see some vesicles. You can even see the mitochondria. Yeah. Okay, so these are what the differentiate between the eukaryotic cells and prokaryotic cells. The major thing is absence of a predefined or a kind of membrane-bound nucleus in the case of the prokaryotic organisms, while in the case of eukaryotic organisms, they possess that membrane-bound nucleus, and they also lack other membrane-bound organelles. Okay. Now, these are the major differences that you see. Apart from what I have just mentioned, in the case of prokaryotic cells, their genetic material or their chromosome that we are familiar with is usually singular, and it is circular in nature. And the ends of the genetic material is attached to one another, so they call it closed. Okay, but in the case of eukaryotic cells, they have chromosomes that are multiple in nature, and normally they are linear. The ribosomes that produces proteins in prokaryotic cells is usually the 70S type, while the eukaryotic cells possess the 80S type. Okay. So now we are going to classify microorganisms. There are two basic classification systems that we will just briefly talk about. We have the earliest classification where the microorganisms or organisms in general are divided into 5 kingdoms. Okay, and we have the recent one where, based on advances in chemistry and molecular genetics, uh, we classify organisms now into only 3 domains. Okay, now for the 5 kingdom classification, we have the fungi, the protista, the monera, planta, and animalia. This is the basic classification we are familiar with when we were doing our basic biology in secondary schools. We say we have kingdom monera, kingdom protista, kingdom fungi, and kingdom planta and animalia. So the 3 kingdoms, the monera, protista, and fungi, are usually where the microorganisms that we are studying in this course are located. While of course the kingdom planta consists of the plants, and of course the animalia consists of the animals, the human beings, and other animals that we are all aware of. Now, because of advances in molecular biology, the classification has been changed, okay, to contain only 3 domains. The first domain is the bacteria, and the second domain is archaea, and the third domain is eukarya. Now, both bacteria and archaea are actually bacteria, but the archaea is an ancient bacteria. The bacteria in the kingdom bacteria contains all those bacterial organisms that are having the true characteristics of bacterial organisms that we know. They are mostly those bacterial organisms that are engaged in causing pathogenic infections in human beings and animals. Okay, so the reason they are called true bacteria is because their cell walls, they possess what is called as peptidoglycan. Okay, so they have peptidoglycan in their cell wall. And even though most of their characteristics between bacteria and archaea are common, they share common characteristics. For example, they are all prokaryotes, meaning that they do not have true nuclear bound membrane. They do not possess nuclear bound organelles. Okay, they all have cell wall, but in the case of bacteria, the cell wall consists of peptidoglycan, but in the case of archaea, the cell wall do not have peptidoglycan, and the cell membrane they possess, they have some unique membrane lipids, which are absent in our normal bacteria. And our normal bacteria are normally found in our basic environment, where human beings and animals usually get in contact with them, and some of the activities they cause as they associate with us, some of it is pathogenic in nature, while some of it is beneficial in nature. But in the case of archaea, they normally are found in extreme environmental conditions, like for example in glaciers and hot springs. That's where you find archaea. They have unusual metabolic processes. For example, there are some bacterial organisms that, based on their own metabolism, they produce methane gas, which is what we can use to generate energy in lieu of the oil we are using. Okay, so those type of bacteria are called methanogens. Okay, they are called methanogens, but there is no any bacteria in in in, in the pathogenic types that usually produce methane gas. Even if they produce it, it's going to be in a very small quantity. Okay, so the third kingdom, where the third domain we have is the eukarya, where most of the eukaryotic organisms are located, and it is made up of protists and fungi. Okay, they are made up of protists and fungi. Now, this protists usually is composed of algae, protozoa, and slime molds, while the fungi consist of yeast and filamentous fungi. The difference between filamentous fungi and yeast is that, as the filamentous fungi is actually multicellular, while the yeast is a unicellular organism. The highest you can see a yeast is when you see a yeast having a bud or a rotor cell attached to it. That is where it is undergoing budding so that it can divide. So you see it appearing as if you have a granite in your hand. Okay, just sometimes when you are eating granite, you can see you may come across one granite that there is one big cell and then small one that is attached to it. So that is how yeast cells usually appear when they are trying to bud. Okay, so most of the algae, they are responsible for producing. They are photosynthetic in nature, and because of their activities, usually you found that they produce most of the oxygen that we breathe in. You know, about 70% of the oxygen that is in circulation in the world is usually produced by algae. Okay, so they are photosynthetic in nature, and you usually find them at the lower level of production within the ecosystem. While for the protozoans, of course, like malarial parasite, for example, we have trypanosoma parasites, which are responsible for causing infection in humans and animals. So some of the protozoans are pathogenic in nature, while some of them are free living. Okay, and for those that are pathogenic in nature, like malarial parasite Plasmodium, and the trypanosoma and Babesia and Ehrlichia, all these are unicellular organisms. So they are protozoans, but they are unicellular organisms. Okay. Now, let us look at the historical aspect of microbiology. We know that we're dealing with microorganisms, and like we said, we cannot see them without the aid of a microscope. Our own resolution, the power of our own lenses that God has given us, cannot see beyond what we can see. So most of these bacterial organisms and fungal organisms that are unicellular in nature, you cannot see them without the aid of the microscope. So definitely the development of the field of the development of the field of microbiology must begin its own history with the time when this become visible, when we begin to see them and know that they are there in existence. Okay, so for that reason, we begin our history with the contributions of two prominent personalities that were able to, or that were referred as the first people to actually start studying microorganisms with the aid of magnifying instruments such as microscope. So Robert Hooke, I'm sure most of us, and Anthony van Leeuwenhoek, most of us are familiar with these names during our basic biology. If you could remember, our first contact with Robert Hooke in biology, we were informed that he used a microscope to view at cork. That cork that he used to stopper bottles. So when he viewed the cork under the microscope, he found that there are certain little boxes that he saw. So he called those little boxes cells. So he was the first person to start defining or to start using the term cell. Okay, so because of that, he kept on studying fungal organisms, and he was actually the first person, the very first person that described or that drew fungal organisms for us to know that they are actually in existence when he was viewing them using the microscope. So he recorded all his findings in his book, or he recorded all his drawings in his book called Micrographia. Okay. Now, the next person that contributed immensely to the development of the field of microbiology is Anthony van Leeuwenhoek. In his own case, since Robert Hooke was the first person to observe fungal organisms and record the way they appear under the microscope, bacterial organisms were actually discovered by Anthony van Leeuwenhoek. Actually, he was a cloth merchant. So he learned uh, grounding glasses to make magnifying lenses so that he can use those magnifying lenses to look at clothes and find defects in fabrics. So apart from that, he decided that let him just further extend the use of his lenses to view at some things. So he got some water and he viewed the water, and behold, he was seeing some tiny animals, moving around. Some of them are motile, some of them are not motile, and they exist in different shapes. So he called them animalcules. animal kills, meaning that small animals. Okay, so when he was observing them, he was drawing what he was seeing, and he related his finding to the Royal Academy of Sciences in London, and that is when they published all the sketches you are seeing. He showed he told them what he has used to see them. So actually, at that particular time, there was no any link for diseases that is affecting animal for diseases that are affecting animals and humans. There was no any link for their microbial origin. Okay, there was no any link for their microbial origin, even though Anthony and Robert Hooke all described fungal organisms, but nobody made the connection between these organisms that they are describing and the nature of infectious disease. Nobody was having that connection between them, that they are actually the cause of infectious diseases. So at that particular time, the concept that was prevalent was the issue of spontaneous generation. Within the scientific community at that particular time, there were people that were having the notion that all those disease conditions that are affecting human beings and animals are actually, caused by bad air. Okay, and they said at that particular time that all living things originate from non-living things. So there was that concept that all living organisms actually develop from non-living matter, meaning that if you take a soil now and keep it, after some time, a living organism will just emerge from it. Okay. So the first persons that were able to start arguing and refuting this claim was Francesco Redi. In his own time, he realized that how can living organisms originate from non-living matter? So he decided to perform some experiments to indicate that no, all living organisms must have had living organism origin. So any living organism you see must have developed from another living organism. So he tried to perform some simple experiments that will refute this. What he did was that he took a meat, two pieces of meat, he put them in two clean separate containers. The first container he covered with a gauze, which will prevent flies from touching the meat. While in the second container, he left it to be open. That will permit flies to be, touching the meat. And as the flies are touching the meat, they deposited their eggs. And eventually maggots developed, from the eggs that the flies laid on the meat that is in the open container. While because they do not have any, they do not have access to the meat that is closed, they couldn't lay their eggs on those meat, and because of that, there was no any maggot developing. But that is on the meat. But because they developed, they laid their eggs on the gauze that covered the container. Eventually, maggots developed on the gauze. So Francesco already now argued that, you see, the reason why maggots developed on the meat that I put inside open container is because the flies were able to lay their eggs on them, and then the eggs hatch, and then you have maggots developing from that. Okay, so that means life begets life. While in the case of the meat that I close with a gauze, you can see that the meat remains maggot free, but the maggots develop on the gauze because the eggs that are being laid by flies on the gauze itself were able to hatch, and then maggot become developed on the gauze itself. So because of that, another person called Louis Jablot, he conducted another experiment. In this time around, he didn't use the meat Francesco already used. Rather, he take hay infusion, meaning that you have a grass that has become dried. That is what is called hay. So that grass that become dried, he now put it in two separate containers. When he put it in two separate containers, he now boiled the hay infusion. Okay. When he boiled that hay infusion, he covered one with gauze, and the other one he didn't cover it with gauze. So the one that he covered, no dust was able to settle on it, and it becomes clear of microbial growth. While the container that he did not cover, despite the fact that he boiled the hair there, because it is uncovered, he want to argue that apart from the flies, even dust particles carry microorganisms that it can deposit on something that is not covered, and that is why you can have microorganisms, developing. So for the hair that he has boiled but uncovered, because of the settlement of the dust in the uncovered container, there was heavy microbial growth there. So he argued that dust particles also can carry, some microbial organisms. They are responsible for that microbial contamination. But when you cover your utensils, where dust will not be able to settle on it, then you will not be able to have any microbial growth. But John Needham, which is a proponent of the theory of spontaneous generation, tried to refute what Francesco Redi and Louis Jablot, proved. He said that he took a mouton and make a broth out of it, and then when he made a broth out of it, he boiled it. When he boiled it, he now kept them in separate flask and covered them. After he covered them, all of them developed microorganisms. There are microorganisms that developed from those, even though he sealed them. So he argued that no, still life can originate from non-living matter, because as you can see, you have argued from your previous two experiments that covering the container will make microorganisms not to develop. But in his own case now, he has covered the containers, and then still microorganisms developed. So he was still happy that he was able to refute what they have really discovered, or what they have tried to use to argue that it is only life begets life. So lest I forget, the theory of spontaneous generation is also called abiogenesis theory, meaning that life originates from non-living matter, while the theory that is supporting that life begets life is called biogenesis theory. Okay, so other scientists now decided to say no. The reason why germs were able to develop from your flask, despite the fact that you boiled it, is that maybe there are certain temperature that the spore form of the bacterial organisms that you have seen actually were not able to be killed by that heat treatment that you have done. That's why, despite the fact that you did that, there was that organism developing. So Franz Schulz and Theodor Swan, all of them are German scientists. They tried to create another, or to do another experiment that will further repudiate the notion that life originates from non-living matter. So they first of all pass an air, because they want to believe that, just like Jablot mentioned, that if you allow your container air to pass through, dust particles will settle in, and that's why you have a microorganism developing. So these two gentlemen, they now pass an air through strong chemicals which are supposed to kill whatever that air can carry. Okay, first it will pass through a strong chemical, and then it will also pass through a heat source. So as the air goes into the container that they are supposed to develop microorganisms, it is supposed to be sterile air, because it has passed through hot heat, and it also has passed through chemical treatment. So that will kill all microbial life that is there. So the air that will eventually reach their sterilized infusion will be a sterile air. So because of that, they were not able they were able to prove that only life begets life, because if that air does not carry any microbial life, when there is no any heat treatment, that means it will be the infusion they have that was sterile will still become sterile. But they know that based on the experiment Jablot performed, they know that that air must have contained microorganisms. That's why they subjected it to this chemical treatment and heat treatment. And if they do that, they know that whatever microbial life that is there will be destroyed. So that's why they said that if that microbial life is destroyed from that air, then that means their container will become sterile. And so it became sterile. There was no any microorganisms that were developing on that. But the proponents of abiogenesis still claim that that rigorous chemical treatment as well as heat treatment must have destroyed the essential ingredients that life requires to develop from. That's why there was no life form that was actually, developed from that sterile broth. So to further make people believe that really there are microorganisms in that air, another scientist, Schroeder and Theodore, they conducted another experiment. In this case, they refused to subject the air to heat or chemical treatment. Rather, they used cotton so that the air will pass through cotton first. And as it passed through cotton, it will now go into that sterile broth. So that cotton plug that they used, was able to eliminate microbial organisms from passing that are inside that air from reaching the sterile broth. So as a result of that, the sterile broth remains germ-free. So despite this, still there are people that are adamant that no, all these things, they still don't believe that actually life does not originate from non-living matter. Despite this proofs. So the final experiment that laid the theory of spontaneous generation, to rest was the experiment conducted by Pasteur. Okay, the first of all, he trapped the using cotton. He trapped air, and that air that he has trapped, dust particles, settle on it, and he observed that later on there are tiny objects that he observed on that cotton that he used to trap the air, and because of the dust particles, he believed that most likely those tiny objects that he was able to see were spores. Okay, so he realized that the same filter that he used to filter an air, with the tiny object that he observed, if he put it inside a broth that is sterile, microbial organisms or microbial growth will ensue. So for that reason, he said that the air and dust must have carried the microbes that cause contamination in previously sterile broths. Okay, so for him to prove this notion, he now used he fashioned some flask, with long neck that is carved. They call it swan neck, after this bird swan. So he will put a broth inside that swan neck, so swan neck flask. He will now heat the broth. When he heat the broth, as long as that swan neck is attached to that particular flask, he will not close that the mouth of the flask with anything. But because it is a swan neck, it's a very long neck, and it is curved, air that is supposed to pass through that will go and contaminate the sterile broth with microbes will not be able to pass through up to the level that it will reach the sterile broth. So for that reason, it will remain sterile. Okay, it will remain sterile. But if he is to break this one neck to make it shorter so that the air can easily pass from the broken part of the neck to reach the sterile broth, then microbial growth will be seen. So because of that, he proved that no, it is the air that contains microbes, that will go and contaminate a previously sterile broth. Okay, and this experiment was what was what was the final proof to indicate that really life begets life. There is nothing like organisms developing from non-living matter. Okay, so apart from this contribution he did, he also contributed in the development of microbiology through some other experiments. There is a process called pasteurization. That pasteurization is what we normally use. Any yogurt now you take, or milk you take, or canned drinks, they are supposed to undergo through the process of pasteurization, which is a kind of short-term treatment of milk so that you can kill tubercle bacilli. You know, there is this Mycobacterium tuberculosis that is responsible for tuberculosis that we are seeing in both animals and humans. So because animals can be infected with this Mycobacterium tuberculosis, the milk you ingest or you take from an animal that is infected with either Mycobacterium tuberculosis or Mycobacterium bovis will have that tubercle bacilli in it. So for you to have a healthy milk that will be wholesome for you to drink, the flavor of that milk will not be lost, but yet the microorganisms that are there will be killed. Is it Mycobacterium tuberculosis or Brucella organisms or any other organisms that can be, that human beings can contract from drinking contaminated milks? When you subject them to the process of pasteurization, the microorganisms there will be killed. Okay, it is just a kind of short-term treatment, a short-term heat treatment to this milk or other canned products that we used to take. Okay. He was also responsible for the development of vaccines against fowl cholera, rabies, as well as anthrax. Okay. So because of that, he became very famous, more especially with the experiments of he conducted using rabies, the rabies vaccine that he was able to develop. So Pasteur Institute he's a French nationalist anyway. So he is a French national. So the French government, they form an institute that is called Pasteur Institute. Initially, the institute actually uh, was made to study or to be producing rabies vaccine. But later on, other forms of vaccines were also, developed in the institute. Okay, so he also postulated a very important theory called germ theory. This germ theory he postulated was the first beginning to associate infectious diseases with microbes. Okay, so he was the first person to say that no, these microorganisms we are seeing are actually responsible for most of these infectious diseases that we are affected with. Okay, so he was considered the father of modern microbiology. Now, because of that germ theory, scientists all over the world now started to say, okay, since Pasteur has, proved that there is that link between microorganisms and diseases that we see in both humans and animals, how can we do, or what can we do to reduce the burden of this microbial organisms so that we will have reduction in infectious diseases? So Joseph Lister is a surgeon. This gentleman, whenever he undergo surgery, he used to find that patients used to die from that surgery. And sometimes, because of the limited number of health personnel, he will go and deliver. He will go and deliver patients in gynecological ward. After taking delivery, he will now go and enter into surgery without washing his hand, without doing anything. And he now realized that as he moved from patient to patient while handling them, actually more maternal death and more death in surgical wards are being recorded. So he was like, okay, since there is this origin of infections as a microbial origin of infections, what can he do to reduce that? So he decided to be using carbolic acid, that is phenol, during surgery and to be dressing wounds in his patient. And he saw a dramatic decrease in mortality in the patients that undergo surgery in his lab. So he was the first person to introduce aseptic techniques, in healthcare provision. Okay. So later on, a German scientist by name of Robert Koch, introduced some postulates known as Koch postulates, where he wants to now further confirm the link between microbial organisms and infectious diseases. So he said that for any organism, to be the one responsible for any infectious disease, then that organism must satisfy these Koch postulates. So the first postulate is that the organism that is suspected to cause a particular disease should be present in all cases of that particular disease, and it should be absent from healthy ones. Okay, it should be absent from healthy ones. And he said that this organism that we are suspecting, which is present in all cases of the disease and absent in healthy ones, must be isolated and cultivated in a pure culture. And when you isolate it and cultivate it in a pure culture, when you introduce it into a healthy animal that is susceptible to that particular disease, they should result in that particular disease. Meaning that isolated organism that we are suspecting, when you introduce it into a healthy animal, then
it's like the facilitator battery is off. You can't hear anything again.
The battery is low. Maybe it's trying to charge it again.
Let go.
We should just hold on. It is coming up.
Okay, better.
Battery module. His lecture. He's a good lecturer.
That's good. Akwụkwọ ụkwa. At last, I was able to log in. Almost the time. Now, is it not 11? Almost 11 o'clock. Thing is annoying.
Congrats.
Thank you. Hey.
Eunice, when the lecturer come back, make sure you mute yourself.
Not Shehu.
All right.
Not Shehu. I think he's not here.
People should mute yourself to the back.
Not Shehu, I don't hear your voice now.
Hello, can you hear me?
Okay, yes.
Sorry, my battery has gone down. I do not have light. Please, can you tell all the rest to put up their mic? They should mute their mics, please. There is too much interference. So the only thing is that we will continue discussing. Hello, can you hear me?
Yeah.
Okay, please, if your mic is on, you should put it off. That is background noise. Benedicta, I can still see you. Okay, so I think the only thing is that we are going to have well, I'm just going to continue discussing because I'm using my laptop to do the lectures, and unfortunately my battery ran down and there is no light now, so we'll just continue discussion from where I stopped. Had it been I put the slides in my phone, I can continue sharing, but I will still discuss. And at the end of the lectures, I will, when there is light, I'm going to put the material for you in the course so that you can all download and see. Okay.
In
So
what,
like I
sir?
said,
In what?
hello, can you all hear me? So
You
like
said you're going to put the material where?
in
I
the
didn't get.
course page.
Yes,
Okay.
sir, no problem. Continue.
Yeah. So, like I said, the coach postulates he was there to he made it so that people will understand the role microbial organisms play in disease processes in humans and animals. So, according to him, all these 5 postulates must be satisfied for you to be able to say that, yes, this organism is responsible for this particular infection. So like I said, he said that one, that particular organism must be present in all cases of that infection or in that disease, all cases of that disease, but it should be absent from all healthy individuals that do not have that disease. And you should be able to isolate them in pure culture. Okay, you should isolate them. You take sample from that disease patient and then isolate them in pure culture. And when you isolate them in pure culture, you should be able to, reproduce the same disease condition when you use that pure culture organism in a susceptible animal or human. Okay, and when that animal developed the disease, you should be able to re-isolate that particular organism from that disease animal or from that disease human being, so that you will see that what you have isolated is actually similar with what you have introduced into that particular
Yes, ma'am.
then that's when you can now beat your chest and say that yes,
Yes, ma'am.
this particular organism is responsible for this infection. But the problem with this postulate is that non-infectious diseases, you cannot prove non-infectious diseases with this postulate. And at the same time, there are certain infectious diseases that they are caused by not one single agent. So any disease that is caused by multiple agents, for example, that particular disease, you cannot prove it using a Koch postulate. But other diseases that are actually having just one etiologic agent, you can prove you can use the Koch postulate to prove the role of that particular organism in that disease process. Okay, so because of that, because of his contribution in medicine in 1905, he was awarded a Nobel Prize in medicine and physiology. Okay? So these are actually the contributions Robert Koch, gave to the field of microbiology. Now, other contributors actually are those that are involved in modern microbiology, the modern era of microbiology, where some of them actually showed the nature of genetic material. Hello, can you please put your mics? Can you mute your mics? There are a lot of interferences, please.
Sir, we are not seeing your screen.
No, no problem.
You cannot see my screen because the slides are actually in my the slides are in my laptop. The slides are in my laptop, and because of that,
Yes, I'm okay. Let's go.
that's why you cannot see me. But I will share the slides so that you can go into the courseware and download them. Okay, so you will find them in topic number 1. You will find them in topic number 1. So this is just going to be a discussion. Now, the next unit we are going to discuss the scope of microbiology. Can you please
Please, people are
mute yourselves?
disturbing. Please, sir, can you mute them centrally so that nobody will hear their voices?
Yeah. Let me mute all. So I think this is okay now. This is okay now. So I have to mute all of you now. So you have to bear with me. Amara, can you please mute yourself? So now what we are going to discuss now is the scope of microbiology. We have been talking about microbiology. What is actually the scope of microbiology? Now, under the scope of microbiology is actually divided into two basic broad fields. We have the basic aspect of microbiology, and we have the applied aspect of microbiology. This course we are offering now is under the applied aspect of microbiology, because the applied aspect of microbiology, deals with those fields of microbiology that normally you use, to aid in human endeavor. While the basic part of microbiology, or the basic aspect of microbiology, is that aspect of microbiology that deals with the study of various components of or various organisms in microbiology. For example, in the basic aspect of microbiology, we have a field known as bacteriology. This particular bacteriology, any person who wants to specialize in bacteriology is a person that will study only bacterial organisms. He's a microbiologist, but he wants to specialize in only bacterial organisms. Okay, we have another aspect called mycology, all under basic aspect of microbiology. This mycology is the study of fungal organisms. Is that field of microbiology that deals with the study of only fungal organisms. So anybody who wants to be a specialist in fungal organisms, he will study the aspect of microbiology called mycology. And in the end, he will be considered mycologist. When you want to study viruses alone, you are going to be called virologist, because you will study that aspect of basic microbiology called virology. When you study bacteria, you are called bacteriologist, okay, because you are only specializing in bacteriology. So there are various fields that are, that are considered the basic aspect of microbiology. When you consider, for example, microbial genetics, where the main aim of this microbial genetics is for you to study the genetic makeup of various microorganisms and the role they play, in disease transmission and what have you. You are called a molecular biologist or microbial geneticist, for example. And that is also an aspect of basic aspect of microbiology. Now, when you go to the applied arm of the microbiology, you are going to be encountering fields like immunology, where you are going to study all those aspects of immunity that a particular patient will mount due to the presence of myriads of microorganisms in their bodies. Okay, and you talk about biotechnology, which is one of the in things now, where you make use of techniques, molecular biology techniques, to improve certain way of life of some microorganisms, so that they can produce some products that will be useful to you. You also talk about industrial microbiology, where, for example, now I want to believe as nurses, at one time or the other, you must have injected one patient or the other having maybe an insulin, more especially if you are in internal medicine. Where diabetic patients is to go, and they will have an insulin injection being prescribed to them. Now, the aspect of biotechnology or industrial microbiology is what is responsible for that mass production of that insulin that you can use to inject your patients with. Okay, So many industrial things now that we use in our homes and in our hospitals are actually the product that are being produced using industrial microbiology. We have another form of microbiology, and that is the microbial ecology, where you study the environment as the interaction between microorganisms and their environment, and what roles they play in disease processes and also in healthy processes. Like, for example, there is an aspect of environmental microbiology that, like, for example, this organic cleaning that is going on. Most likely what they are using to clean up that environment now is the aspect of environmental microbiology, where some bacterial organisms are used that have been genetically modified to actually use up the oil spillage as their food. So by taking off the oil that is being spilled in that environment, and they converted they convert it to energy for their own use, they will clean up that environment. So you can see we are applying microbiology now, to solve environmental problems. So that is another aspect of microbiology. We also have agricultural microbiology, where the microbes are used, to produce insecticides and pesticides, and also these microbes interact with plants, to make some nutrients available for those plants, and the soil content or the nutrient content of the soil, to be enriched as a result of their activities. So that is an aspect of agricultural microbiology. And of course, there are many other fields, that usually we normally have, as a branch of microbiology that we'll take care of, which will make human life easier. So these are the basic aspects of microbiology that you should be aware of. We have the basic arm of microbiology, and we have the applied arm of microbiology. So as nurses, you are going to be interacting with patients, and you're expecting to be giving them care, give them quality care. So your own aspect of microbiology is an applied aspect of microbiology. That's why we said the medical microbiology you see is part of, or is an applied aspect of microbiology. So that's why you are taking this course. We want you to know about these microorganisms and how you can, when you know them, when you know their biology, when you know their weaknesses, you know what you can use to treat them. You know how you can use to the methods you can use to prevent infection. You will be able to deliver quality care to your patients as you encounter them in your practice. You will also be able. Okay, so that is the end of this lecture. It's supposed to be a 1 hour lecture. So I think when you want to, if you have a question that you want to ask, you can ask by first raising your hand. So if I mention your name, you can just unmute your mic, and then so Benedicta, you can unmute your mic and ask your question, please.
Good morning, sir.
Good morning, Benedicta. How are you?
I'm fine, thank you. So I want to thank you so much for the lecture. You have really made it simple for us. My question is on Robert Koch's first postulate that say that the disease-causing organism should be present in all cases of the disease and absent from healthy animals. So how then can we reconcile the concept of healthy carriers?
That is also another aspect that the Robert Koch postulates were not able to put into consideration. Okay, because as far as he's concerned, during his time there was no knowledge of healthy carriers, that was known then. So because of that, he was only using the knowledge that was available to them at that particular time. So this is part of the limitations of the Koch postulate. This is part of the limitations of the Koch postulate, because that's why I said all those disease conditions that have multifactorial or multi-agent causes, he cannot account for them. For example, now if you use, for example, the concept of Koch postulate, now you cannot prove AIDS as a condition using Koch postulate, because apart from the virus, there are other secondary bacterial and other fungal organisms that can complicate, a HIV patient life to become an AIDS patient, isn't it? So you cannot use that particular postulate to prove it. So it's one of the limitations of Koch postulate. So it is only in modern times now that we are able to know that there are certain people that are, harboring an infection, even though they are looking apparently healthy, but they are actively spreading that infectious agent to other people in the environment that are living together with them, and those people can become, infected with that particular agent that the carriers are taking. So all forms of carriers, be it recovery carrier, healthy carrier, those that are in early phase or latent carrier, the Koch postulate cannot actually take care of them. So that's one of the limitations of the Koch postulate. And to take care of these postulates, normally there are modern postulates that has been, that has been postulated. They're almost similar to the Koch postulate, but they are using genetic material, to prove the role of an organism in disease processes. So in such situations, you find out that even those people that are healthy, they will be sampled. And if that agent is there, once it is genetic material can be identified and they sequence it and they know that it is there, they know that yes, this agent is responsible for this, even though this is a healthy carrier. So the modern Koch postulate is what will take care of your question. That is the aspect of healthy carriers.
Okay. Thank you, sir.
Fidelia, you're welcome, Benedicta. Fidelia, can you hear me? You raised your hand.
Good morning, sir. Thank you for the lecture this morning.
Good morning, Fidelia. Thank you.
I want to find out, when we were registering this course, there was no course material for it. They said it's a new course, so there's no course material yet. So how do we get the material to use for the course?
Actually, I'm actually reviewing the course material. But this one that the course material that you can get for this course. Hello.
Hello, sir.
Sorry, let me mute all of you because there is interference too.
Yeah, so much noise everywhere.
Let me. So can you hear me, Fidelia?
What is GOPD? GOPD.
Fidelia. Can you mute your mic? Thank you. So Fidelia, to answer your question, when you go to the noun course material, noun courseware, when you type noun courseware in your distance, it will take you to a kind of repository where all the course materials are there. So when you go there, you select in the field, you select in the faculty, you select health sciences. So once you select health sciences, you will see a course called NSC211. You will see that course material there, so you can download it. So that is what we are now using, to teach this course. I'm actually the one reviewing the course, so there are some updates that I will be able to that I'm doing to the course material. So later on, when they approve it, it will be made available to all of you. But as at now, what you can use is what is going to be available to the course material. And I'm actually teaching you based on what is there in the e-courseware. So when you put in your browser noun e-courseware, it will take you to that page. So you just go, you see some fields that you should impute. So in the field where they said faculty, you put health sciences. Once you put health sciences and you click, you will see various course materials. As you scroll down, you'll see various course materials that will be available to you. And you just look at NSC211 medical microbiology and download it. That is the course material we are using currently. I hope I have answered your question. Chiamaka. Chiamaka, if you can hear me, you just unmute yourself and ask your question. Chiamaka. Okay. Ekubi.
Hello, sir. Good morning, sir.
Yeah. Hello, good morning. Okay, Chiamaka, you first. Hmm. Is it Chiamaka speaking now?
Yes, Chiamaka speaking.
Okay, go on.
Thank you for the lecture, sir.
You're welcome, Chiamaka.
Sir, please, I want to know who is the father of microbiology.
Well, when you say the father of microbiology, usually there are about 2 or 3 fathers. The first person that was able to observe fungal organisms, to know that there are actually microorganisms called fungi, it was Robert Hooke. It was Robert Hooke. So you can call Robert Hooke the father of mycology, which is the study of fungal organism, because he was the first person that described their existence. Now, if you are talking about bacteriology now, you can say Antonio van Leeuwenhoek is the father of bacteriology because he was the one who first described bacterial organisms.
Okay, sir.
Okay, so you see we have 2 fathers now coming up, right? For mycology and for bacteriology. Now, if you want to say the father of virology, for example, then you are going to talk about these 2 guys, Bergering and Frosk.
Okay.
And when Bergering and Frosk were studying what is called the tobacco mosaic virus, they were the first people to actually relate infectious viral diseases to indicate the infectious nature of viral diseases, because tobacco mosaic virus was the virus causing economic losses to the tobacco industry at that particular time. So they did some series of experiments to indicate that actually a filterable agent that is capable of independent replication is responsible for that particular disease condition. And almost at the same time, Frosk, he and his colleague also indicated the infectious nature of foot and mouth disease condition in animals. So this, in case of virology, you can say they are the father of virology. Now, in case of immunology, because of vaccination, you can say that Edward Jenner is the father of immunology because he was the first person to use a process known as variolation to protect people that are coming down with smallpox infection using cowpox virus. So he used a kind of a related virus that is causing disease in animal, and he used it to infect human beings so that they will come with mild type of that disease. So when they come with that mild type of that disease, they later on become protected against catching smallpox, which is very deadly. So in the case of vaccinology or immunology, you can say Edward Jenner is the father of immunology. But when you talk about modern microbiology, then Louis Pasteur is considered the father of modern microbiology, because he was the first person that introduced that germ theory. And it was on the basis of that germ theory that he said all infectious diseases have microbial origin. It was on the basis of that, tried to use their knowledge of the germ theory proposed by Louis Pasteur to say that, yes, what can we do to prevent infection? In case of Joseph Lister, he was the first person that introduced that aseptic technique in hospital setting using phenol as disinfectant. So the whole process of disinfection started with Joseph Lister. And say that this organism is responsible for this disease, to confirm that actually it was based on the Koch. So it all depends on what context you are talking about. But normally when you see in some textbooks, they will tell you that the father of microbiology, they will say Anthony van Leeuwenhoek, because he was the first person to describe bacteria. But that does not eliminate the contribution of Robert Hooke, who actually, studied fungal organisms using his own microscope. So that's it. So I hope I have answered your question.
Yes, sir. Thank you, sir.
Okay. So, it could be.
Yes, sir. Good morning, sir.
Yeah. Good morning.
Yes,
So you
sir.
can ask your question.
Mic. Noise.
You said? Sorry?
I mean, I think the noise is coming from Shamaka's point. She mutes it so that we'll all hear.
Okay. Chiamaka, please mute your mic.
Okay, sir, please. My question is, I wanted to find out, because concerning the course material, somebody asked earlier.
Okay.
The NSC211, we discovered that we have a similar one in the hard copy, which was the former NSC106. So as we are going along with your lectures, for now is the same with what we have. And so what I want to find out, sir, is that if this course has been reviewed and maybe some things will not be added in the hard copy or some things will be out, so that we know to fall back to the soft copy. Because some of us don't have the soft copy, but we are making use of the hard copy. So should we still continue on it? Is it the same?
Yeah, continue using it for now. Like I told you, I'm the one reviewing the course material, and there are some additional things that I have imputed that are not here. But the head of department told me that I cannot use it now to teach you because it has to undergo some administrative processes for it to be approved before it will be uploaded to you. You understand? So as we go, all those things, even if we are done with our lectures, as soon as they approve this one, I will organize 1 or 2 lectures to cover all those aspects that I have added, which we did not cover.
Okay, sir. Okay, thank you very much. I'm grateful.
You're welcome.
Okay, sir. Please, immediately you upload it, let us know so that we'll see.
Definitely, I will let you know.
All right, thank you very much. I'm grateful.
You're welcome. You're welcome. So, any more question? I have seen that 2 more people that raised their hands earlier were not. They all lowered their hands. Are we all okay with the responses so far? Okay. Chinyere, yes. Please unmute yourself and ask your question.
Good morning, sir.
Good morning, Chinyere. How are you?
I'm fine, sir. Thank you very much for your lecture, sir. It's
You're welcome.
Yes, sir.
Okay, thank you.
My question has been answered. I raised my hand initially, but I wanted to ask this particular last question that she asked.
Okay.
So
Okay.
Thank you,
okay.
sir.
Okay, you're welcome. You're welcome, Cherry. Thank you. So I think in the absence of anything, this marks the end of our lectures for today. So we'll meet next week, same time. And I hope it's going to be another fruitful discussion with you. Thank you all very much. And I wish you a fruitful day. Thank you.
Thank you. Same time, same day.
Oh, yeah, same time, same day. Yes.
Okay.
Thank you, sir.
Thank you, sir.
All right. Thank you.
Thank you,
very
sir.
much. Thank you.
Yeah.
Happy Independence, sir.
All right. I'll send to you all.
Thank you all.